Okay, so today we are going to see nucleic acid part from your biochemistry. We have already seen carbohydrate, proteins, lipids, their metabolism, amino acids, vitamins, and all important points from those chapters. So only one section is remaining from your biochemistry, and that is nothing but the nucleic acids. So nucleic acids. First of all, you should know what do you mean by nucleic acids. As the term indicate, उसके नाम में ही है न्यूक्लिक एसिड मतलब क्या है जो न्यूक्लियस में मिलता है वो एसिड न्यूक्लियस में कौन सा एसिड मिलता है दैट इज नोन एज अ डीऑक्सीराइबोन्यूक्लिक एसिड राइबोन्यूक्लिक एसिड देयर आर टू न्यूक्लिक एसिड्स they mainly serve as a repository for your genetic information or they are a hereditary information will be transmitted from one generation to another generation by the means of nucleic acids. So what are the nucleic acids? First is remember the nucleus. It is made up of two terms, nucleus and acids. So what is nucleus? Nucleus that occurs in the cell. Nucleus कहाँ पे होता है? Cell में होता है. So उसके अंदर जो acid है उसको बोलेंगे nucleic acid. So there are two kind of acids present in the nucleus. So what are these? They are nothing but the first one is a deoxyribonucleic acid and it is also known as a DNA, deoxyribonucleic acid. And second one is nothing but the RNA, ribonucleic acid. Okay? So deoxyribonucleic acid, ribonucleic acid. These are the two types of nucleic acid. They are the very important for a so everybody has different genetic makeup. Everybody has different body shape. Everybody has different hormones. Everybody has different enzymes and all that. Okay. So all these the shape, proteins, hormones, okay, external shape, the physique of your okay, that is decided by your genetic makeup, and that is nothing but the information that is stored in the DNA. That is a deoxyribonucleic acid, ribonucleic acid. These two are the main important nucleic acids. Okay. Now DNA was discovered by this Frederick Michener. Okay. Remember DNA was first time discovered by Frederick Michener. Then this DNA consists of a genetic material and it was demonstrated by carrying out one experiment. तो DNA पहली बार जो फाइंड आउट किया था डिस्कवर किया था उसका नाम है फेडरिक मिशनर ओके मिशनर फेडरिक मिशनर डीएनए में जेनेटिक इंफॉर्मेशन होता है या फिर डीएनए में हेरिडिटरी मटेरियल होता है तो ऐसा डेमोन्स्ट्रेट किया किसने experiment was carried out in 1944 by Avery, McLeod and McCary. So they demonstrated that DNA consists of a genetic information. So then what is the DNA? It is nothing but the chemical basis for hereditary and genetic information. So it is a chemical basis for genetic information and hereditary characters. This genetic information it is organized into genes. Okay, so DNA that is deoxyribonucleic acid is organized into genes. तो आप जब बोलते हैं कि इसके जीन में है उसके जीन में है so gene are nothing but the nucleic acids. The DNA is the very basic nucleic acid and it is organized. It is packed in the gene. Okay, so gene is the you can say it is a storage out of DNA, which is the fundamental unit of your genetic information. So gene is nothing but the fundamental unit of your genetic information, and in that gene you have DNA. The genetic information it is stored on the DNA. Okay. Now the relation between this DNA, then RNA and protein. So interrelationship between DNA, RNA and protein that is known as the central dogma of lab. Okay, central dogma of lab or it is a central dogma for molecular biology. तो ये सेंट्रल डॉगमा ऑफ मॉलिकुलर बायोलॉजी और सेंट्रल डॉगमा ऑफ लाइफ मतलब क्या कि जो डीएनए है आपका आरएनए है और प्रोटीन से प्रोटीन्स एंजाइम्स आल्सो प्रोटीन्स तो whatever the relationship between DNA, RNA and protein that is known as a molecular dogma okay or central dogma of life. So molecular dogma of biology basically every individual cell has a DNA. हर एक सेल में डीएनए होता है so that DNA makes its multiple copies duplicate copies known as a daughter DNA. So making of copies of DNA is known as a replication and after that replication making of RNA ribonucleic acid from DNA that is known as a transcription. Synthesis of protein that RNA is known as a transcription. Replication, transcription, translation. These three processes they are known as a central dogma of life. Remember, okay? Now these DNA and RNA, deoxyribonucleic acid, ribose nucleic acid, they are nothing but the polymer. Okay, as like your other biomolecules. जैसे होता है कि कार्बोहाइड्रेट में क्या है, पॉलीसेकेराइड इज अ पॉलीमर ऑफ मोनोसेकेराइड, proteins are the polymers of amino acids. So likewise that nucleic acid are also a polymer. So it is a polymer of nucleotides. So they are the polymer of nucleotides. Nucleotides, okay. So polymer of nucleotides. So nucleotides are also of different type. Like for example, deoxynucleotides, ribonucleotides. So DNA is a polymer of deoxynucleotides, and RNA is the polymer of ribonucleotides. So what is the nucleotides? So nucleotide is a it is a it consists of three parts. So यहाँ पे इसके ऊपर क्वेश्चन आ चुका है न्यूक्लियोटाइड्स. न्यूक्लियोटाइड्स इसमें टी आता है, यहाँ पे टी आता है न्यूक्लियोटाइड्स. न्यूक्लियोटाइड्स कंसिस्ट ऑफ, so remember it is a T for three. T for three. Three means what? Three components. And what are these three components? Remember T for three. Nucleotides जहाँ पे टाइड आ गया, टी आ गया, वहाँ पे there are three components. So first component is nothing but the nitrogenous base. First one is a nitrogenous base. Second component is a pentose sugar, and third component is a, so these three nucleotide consists of three parts. Nucleosides consist of only two parts. So nucleotides are nothing but the nitrogenous base plus pentose sugar plus phosphate group. Now this nitrogenous base means they might be either adenine or guanine. Sorry, purines or pyrimidines. They may be either purines or pyrimidines. This nitrogenous base means purines and pyrimidines plus pentose sugar. So pentose sugar and this nitrogenous base, either purine or pyrimidine. So they are bound together by N glycosidic bond. They are bound together by N glycosidic bond. Pentose sugar plus nitrogenous base. That is nothing but the purines or pyrimidines. So they combine with to form a N glycosidic bond. Again, this phosphate group is attached to this nitrogenous base plus pentose sugar by phosphodiester bond. By phosphodiester. So these are, there are three components of your nucleic acid. Okay. So nucleic acid itself is a polymer. Polymer of what? Nucleotides. So nucleotides is made up of nitrogenous base plus pentose sugar plus phosphate. Okay. याद रखो, क्वेश्चन आ चुका. Now another term is there, nucleosides. Nucleosides without phosphate group. Okay. Only nitrogenous base and pentose sugar. They are the nucleosides. न्यूक्लियोटाइड्स मतलब तीन, साइड्स मतलब दो. साइड्स मतलब ओनली दो जो है, उसमें नाइट्रोजन बेस रहेगा और उसमें पेंटो शुगर रहेगा. न्यूक्लियोटाइड्स मतलब थ्री, पेंटो शुगर प्लस फॉस्फेट ग्रुप प्लस नाइट्रोजनस बेस. All these nucleic acids, they are a polymer of nucleotides. So your DNA is a polymer of deoxynucleotides and RNA is a polymer of ribonucleotides. Okay, मतलब DNA में deoxyribose sugar रहेगा जो पेंटो शुगर होता है. So pentose sugar present in the DNA, that is a deoxyribose sugar, and in the RNA it is a ribose sugar only. What is ribose sugar? Deoxyribose sugar. I will explain. There is only one difference of uh, hydroxyl group. Okay, I will explain in the upcoming slide. Nucleotides, they are nothing but the monomers. They are nothing but the repeating, or they are nothing but the building blocks for DNA and RNA. So in that you have nitrogenous base. So it is made up of either purines or pyrimides like adenine, guanine, okay, adenine, guanine, thymine, cytosine, uracil. So adenine, guanine, thymine, cytosine, uracil. These are nothing but the nitrogenous bases. Okay. So depending on their chemical structure or depending on the heterocyclic ring present in these nitrogenous bases, there are two types. Okay. First one is the purines. So purine में कौन होता है, याद रखो. Purine consists of two, adenine and guanine. Purine consists of two, that is adenine and guanine. तो adenine guanine क्या है? Purine है. तो pyrimidine कौन है? Pyrimidine में देखो cytosine, thymine and uracil. Okay, cytosine, thymine and uracil. So purines में आता है adenine, guanine. Pyrimidines में आता है cytosine, thymine and uracil. So remember in the DNA में क्या होता है? So DNA consists of same DNA and RNA both consist of सेम प्यूरिन्स. प्यूरिन्स क्या होंगे डीएनए में और आरएनए में, दोनों में सेम होंगे, लेकिन पिरिमिडिन्स क्या होंगे, थोड़ा सा अलग होंगे. सो प्यूरिन्स इन द डीएनए एज वेल एज आरएनए दे आर सेम. ओके, बट इन द आरएनए, ओके, व्हाट इज द डिफरेंस? So DNA RNA differs in the pyrimidine base. Purines उसमें सेम रहता है, pyrimidine difference रहता है. Particularly, so in place of thymine, your RNA का stop है uracil. So DNA में thymine रहेगा, ये pyrimidine, और RNA में क्या रहेगा, uracil. So DNA का stop है thymine, purine, sorry pyrimidine, and RNA का stop है uracil. That is the difference. Sugar part of your nucleotides, that is nothing but the pentose sugar. Most commonly is nothing but the ribose sugar. Okay, so D ribose sugar is present in the RNA. राइबोन्यूक्लिक एसिड में डी राइबोस शुगर रहेगा, डी ऑक्सीराइबोन्यूक्लिक एसिड में डी ऑक्सीराइबोस रहेगा. तो दोनों को भी कॉन्फिगरेशन क्या है, डी है, डी नॉट अ एल. तो डी राइबोस एंड डी ऑक्सीराइबोस डीएनए एंड राइबोस आरएनए. These are the types of sugar present in the nucleic acid, and they are bound to a nitrogenous base by which kind of bond? Beta N glycosidic bond. तो आप देखो शुगर और कोई शुगर और जो नाइट्रोजनस पार्ट होता है, जैसे ग्लाइकोसाइड में होता है शुगर नॉन शुगर पार्ट, एक ग्लाइकोन will form a glycosidic bond. So you can correlate that this nitrogenous base is nothing but a glycon part, and that sugar part is a glycon. So bond formation is nothing but the beta N glycosidic bond. So you should remember यहाँ पे 2 या 3 क्वेश्चन बन सकते हैं. So first one is nothing but the who discovered the DNA, that is a Frederick Miescher. Okay, Frederick Miescher. And that DNA cast of a genetic material was demonstrated by McLeod, okay, Avery, McLeod and McCarry in 1944. Then again you should remember what is meant by nucleoside, what is meant by nucleotide. न्यूक्लियोटाइड क्या होता है, न्यूक्लियोसाइड क्या होता है, इसके ऊपर ऑलरेडी क्वेश्चन आ चुका है. सो क्वेश्चन वाज लाइक दैट कि न्यूक्लियोटाइड कंसिस्ट ऑफ फॉलोइंग, न्यूक्लियोटाइड कंसिस्ट ऑफ फॉलोइंग. ऑप्शन ए में था, ऑप्शन ए में रहेगा आपका उसमें नाइट्रोजनस बेस प्लस पेंटोस शुगर, नाइट्रोजनस बेस. ऑप्शन बी नाइट्रोजनस बेस प्लस फॉस्फेट ग्रुप. ऑप्शन सी रहेगा नाइट्रोजनस बेस प्लस पेंटोस शुगर प्लस फॉस्फेट ग्रुप. ऑप्शन डी में रहेगा ऑल ऑफ द अबव. So they will confuse you. So you should be clear. तो इसीलिए टाइड मतलब क्या है, टी जहाँ पे आएगा वो उसमें 3 रहेगा, टी फॉर 3. सो न्यूक्लियोटाइड्स मतलब नाइट्रोजन बेस पेंटोस शुगर एंड फॉस्फेट ग्रुप. साइड्स मतलब 2. साइड कितने होते हैं, 2 ही साइड होते हैं ना, एक तो आगे वाला या पीछे वाला है ना, 2 साइड होता है, लेफ्ट साइड या राइट साइड. सो साइड में क्या कितना रहेगा, 2 ही रहेगा. 2 का क्या रहेगा, नाइट्रोजन बेस 1 रहेगा, पेंटोस शुगर 1 रहेगा. So sides मतलब 2 रहते, टाइड्स मतलब 3 रहते. So depend on that they are either purines and pyrimidines. So purine most commonly found purines in the nucleic acid. कौन से purines होते हैं मोस्टली, adenine and guanine. Okay, pyrimidines कौन से होते हैं मोस्टली, cytosine, thymine and uracil. Now DNA and RNA डिफरेंस क्या है उसमें, एक तो पहला डिफरेंस है आपका शुगर. So in the DNA you have deoxyribose sugar, in the RNA you have ribose sugar. First second difference in the DNA you have, these pyrimidines like cytosine and thymine. Okay, cytosine and thymine, these are the pyrimidine bases present in the DNA. But in the RNA, in place of instead of this thymine, okay, you have uracil. In case of RNA you have uracil. Okay, these are the 2 main differences. And third difference is nothing but the DNA most of the time DNA is a double helical. Okay, DNA is a double helical, and most of the time RNA is a single stranded. So DNA is a double stranded, okay, most of the time, and RNA is a single stranded. But RNA single stranded होता है most of the time. तो अगर उसमें कुछ बेस पेयर में जैसे होता है, RNA का स्ट्रक्चर स्टेबल है, that is because of the adenine. So structure of RNA is stable because of the adenine. Now structure of RNA is a single stranded, and that is stabilized by adenine. So if adenine is replaced by some other bases, what will happen? The rigidity of the structure will be lost. So it may bend and there may be interaction. So at that time it may become a double stranded. Okay, but most of the time DNA is double stranded and RNA is a single stranded. You have to remember. Okay, then most commonly found sugar in this nucleic acid, it is nothing but the pentose sugar, and that probably is a ribose sugar. So D-ribose. Okay, D-ribose sugar is found in the RNA, and deoxyribose sugar is found in the DNA. Remember, and this nitrogenous base and sugar, they are bound together by this glycosidic bond, बीटा एन ग्लाइकोसिडिक बॉन्ड. What is the nucleoprotein? Nucleoprotein मतलब क्या? It is a complexes of protein with the nucleic acid. Complex of protein with the nucleic acid. That is known as a nucleoprotein. So it consists of a nucleic acid plus protein. So nucleic acid is wrapped in the proteins. तो न्यूक्लिक एसिड के ऊपर एक प्रोटीन का कवर रहेगा, डीएनए के ऊपर क्या रहेगा? कवर रहेगा. ओके, so nucleic acids they are wrapped in the cover, and that proteins are known as either histones or protamine. तो ये दो टाइप के प्रोटीन होते हैं न्यूक्लिक एसिड के ऊपर. So those constitute your nucleoprotein. Now if you digest this nucleoprotein or if you hydrolyze this nucleoprotein, then you are getting nucleotides. You are getting nucleotides. Now how you can digest this nucleoprotein, or how you can break down this nucleoprotein? So you can break down this nucleoprotein by two ways. First, you can digest this nucleoprotein by using various enzymes like nucleases. Okay? You can digest by nucleases like polynucleases, like ribonucleases or deoxyribonucleases. They will hydrolyze the phosphodiester bond in the nucleoprotein. Now what will happen? You will get a nucleotides. Again, अगर आप nucleases नहीं use करना चाहते तो you can digest this nucleoprotein by using acid hydrolysis, sorry, base hydrolysis or alkali hydrolysis. Okay, so aqueous ammonia you have to use at 115 degrees. My point is nothing but the hydrolysis of this nucleoprotein will give you nucleotides, and again hydrolysis of this nucleotide will give you nucleosides plus phosphoric acid. So how you can hydrolyze this nucleotides? तो इसको हाइड्रोलाइसिस करने के लिए कौन लगेगा? इसको लगेगा मोनोन्यूक्लियोटाइडेज. ओके, मोनोन्यूक्लियोटिडेज या फिर फॉस्फेटेज, मोनोन्यूक्लियोटिडेज और फॉस्फेटेज. So they will digest nucleotides. They will hydrolyze the ester bond present between sugar and phosphoric acid. So sugar will be released, phosphoric acid will be released, and only nitrogenous base plus sugar will be released. That is known as a nucleosides. तो नाइट्रोजनस बेस शुगर क्या है? दो ही है, तो साइड कितने होता है? दो. ओके, so phosphoric acid will be released. Now again you can again also digest this nucleotide by using, now nucleotides they can be digested by this enzyme known as a nucleosidase, purine or pyrimidine nucleosidase. They will digest nucleosides to release the free nitrogenous bases, that is nothing but purines or pyrimidines. And sugar कौन से होते हैं? D-ribose or D2-deoxyribose. So depending on nucleoprotein, nucleoprotein में अगर DNA है आपका, nucleoprotein में अगर DNA है तो क्या मिलेगा आपको? यहाँ पे purines, pyrimidines प्लस यहाँ पे deoxyribose शुगर मिलेगा. अगर आपके nucleoprotein में सिर्फ RNA है तो यहाँ पे क्या मिलेगा आपको? Ribose शुगर मिलेगा. So these are nothing but the purine and pyrimidine bases. So this one is a purine base. First one is a purine base. Numbering will start from, so this one is a 1, this one is a 5, 6, 7, 8 and 9. That's it. So ऐसा numbering 1, 2, 3, 4, 5, 6, 7, 8 and this one is 9. 9. So example of purines are adenine and guanine. क्या example इसके purine के? Adenine, guanine, they are the most commonly occurred purines in the DNA and RNA both. तो यहाँ पे याद रखो DNA और RNA में purines क्या रहेंगे? सेम रहेंगे. pyrimidines में डिफरेंस है. यहाँ पे pyrimidines में DNA और RNA में डिफरेंस है. So I will explain this. तो टाइम पे आते जाओ, इसलिए आपको लेट लिया है. नॉर्मली 5 बजे होता है, यहाँ 6 बजे इसलिए है कि ज़्यादा से ज़्यादा ज्वाइन हो सके. Now this is the structure of your guanine. Guanine is a type of adenine. This carbon is 1. You can say 3 amino 5 oxopurine, that is nothing but the guanine. Okay, and this is the adenine का structure थोड़ा सा wrong हो गया. So this part should be from this side, यहाँ पे होना चाहिए. So again 5 amino purine, that is nothing but the 6 amino purine, that is nothing but the adenosine. 6 amino purine is adenosine, and here 2 amino 5 oxopurine, that is a guanine. Okay, so these are the two commonly found purines in the DNA और RNA. न्यूक्लिक एसिड में ये दोनों purines होते हैं. pyrimidines कौन से होते हैं? pyrimidine देखो ये it is a structure of pyrimidine. So 1 3 nitrogen. Cytosine, thymine and uracil. Cytosine, thymine and uracil. These are the 3 pyrimidine bases found in the nucleic acids. Nucleic acid मतलब both आएंगे आपका DNA RNA. But in the RNA, in place of thymine, you are getting uracil. You can say in the RNA, adenine guanine is there. Sorry, in the DNA, adenine guanine type of purine is there. So these are the most commonly found purines in the DNA as well as RNA. Now in the DNA, cytosine and thymine. In the DNA, cytosine and thymine, these are the most commonly found pyrimidines in the DNA. In the RNA, this cytosine and uracil, these are the two types. Okay? There is a absence of thymine, okay, in the RNA. As you can see over here, so ribose sugar and deoxyribose sugar. The difference between ribose sugar and deoxyribose. So this is a pentose sugar. ये 5 कार्बन आटम वाला sugar है कौन सा ribose sugar. So you can see 5 carbon 1 2 3 4 5. Okay. D-ribose sugar, it is a structure of D-ribose sugar. Now what is the difference between deoxyribose? So 2 D2 D2 मतलब deconfiguration होता है डेक्स्ट्रो है दोनों. 2 deoxyribose 2 deoxy मतलब यहाँ पे ऑक्सीजन नहीं है deoxy. Okay. That is the difference. 2 D-ribose and D2 deoxyribose. That is the difference. What is meant by central dogma of life? So making of identical copies of DNA. Making of identical copies of DNA. There is a replication. Making of RNA or synthesis of RNA from DNA. That is a transcription. Synthesis of proteins from RNA. Synthesis of proteins from RNA. That is a translation. So interrelationship or the relationship or interlinking of DNA, RNA and protein. That is known as a central dogma of life. Okay? This is the structure of your DNA. तो DNA will look like this. DNA का स्ट्रक्चर कुछ इस तरीके से रहेगा. तो उसको बोलते हैं ट्विस्टेड लैडर. DNA स्ट्रक्चर will look like a twisted ladder. लैडर मतलब आपका जो सीढ़ी होता है तो ट्विस्टेड लैडर इससे आपका वैसे रहेगा. Now as I have told you, DNA, RNA, okay, we'll talk about the DNA first. So it is nothing but the polydeoxyribonucleotide. It is a polymer of deoxyribonucleotide. यहाँ पे deoxyribonucleotide का polymer है DNA. Okay? So DNA has a two chains. DNA has a two chain of okay. So two chain of polydeoxyribonucleotide. So this is chain one. This is chain two. They look like a twisted ladder. This chain, they are two chains. So they are not identical. ये दोनों identical chain नहीं होते. So they are the complementary to each other. Okay? They are complementary to each other. Complementary मतलब वो opposite रहेगा. Okay? So they are the complementary. Now first is one chain, any one chain out of these two. One chain is known as a leading strand. Is known as a template strand or leading strand. तो दोनों एक ही है. Template strand or leading strand, both are the same. So one polynucleotide chain is known as a template or template strand. So on that you have genetic information. तो इसमें दो चेन है आपको देखो. तो एक चेन को बोलते हैं टेम्पलेट स्ट्रैंड, एक को बोलते हैं कॉम्प्लीमेंट्री स्ट्रैंड. जो टेम्पलेट स्ट्रैंड होता है उसके ऊपर आपका genetic information उसके ही ऊपर रहता है. So template strand will have genetic information in the DNA. Complementary strand will not have. Okay. Leading template strand is also known as a leading strand, and this okay, complementary strand is known as a lagging strand. Leading and lagging याद रखो इसको. टेम्पलेट मतलब लीडिंग, लीड कौन करेगा, टेम्पलेट वाला करेगा, और लैगिंग ये. नाउ द स्ट्रक्चर ऑफ, so whatever this nucleotides are there, okay? So purines and pyrimidine bases, not nucleotide, purines and pyrimidine bases. So they may show keto-enol formation. जो purines होते हैं, pyrimidines होते हैं, उसमें keto-enol formation हो सकता है. You can see over. So it is a keto form of your cytosine. Keto form of your cytosine. यहाँ पे ketone group है. So it is known as a lactam form. It's known as a lactam. When ketone group and nitrogen, okay, NH, okay, ये जो है पूरा, this is alpha. This is alpha 2. इसके साथ में जो कार्बन इसको alpha बोलेंगे. When ketone group and amino group they are together, their position is alpha. They are known as a lactam. What is lactam? So you might have seen the beta lactam arrangement. उसमें भी देखो same arrangement होता है. एक में NH होता है और दूसरे बाद में C double bond होता है. That is the lactam arrangement. Okay? So lactam form of your cytosine. So it is a keto form. Now in the solution they may be converted into enol form. Okay? enol or lactin form. So lactin form of your cytosine. You can see over here, यहाँ पे ketone नहीं है, यहाँ पे क्या है, alcohol है. So keto-enol formation. Purines, pyrimidines, they may show keto-enol formation. How there is a bonding between your purines, pyrimidines and your ribose sugar. Okay. As you can see here, adenine. Okay, it is a structure of adenine. Okay, adenine है. A 6 amino purine. 6 amino purine is nothing but the adenine. Okay. So the ninth carbon atom is उसके ऊपर जो है, that is nitrogen. So it will form a bond with the first carbon atom of your ribose sugar. Okay, so this is the first carbon of ribose sugar. So first, second, and then fourth, and this one is a fifth. So that's why this known as a 5 dash. It is known as a 5 dash because how you can define the bonding between two ribonucleosides. two nucleotides रहेंगे तो उसमें ये 5 dash. So 5 dash के ऊपर क्या अटैच होता है? 5 dash के ऊपर अटैच होता है आपका फॉस्फेट. So there will be the formation of phosphodiester bond at the 5 dash end of this nucleoside. So this is a nucleoside. It is not a tide. ये टाइड नहीं है, ये साइड है. क्यों? इसमें दो ये दो साइड होता है. दो nucleoside में there are two components. First one is the nitrogenous base and second one is your pentose sugar. Okay? So at this fifth carbon atom known as a 5 dash, there is a formation of diester bond, phosphodiester bond. यहाँ पे phosphate अटैच होगा बाद में, and ये क्या बनेगा? न्यूक्लियोटाइड बनेगा. So यहाँ पे बीटा आयन ग्लाइकोसाइडिक बॉन्ड. Now again central dogma of life. So आपका जो DNA है, so your cell will replicate the DNA. It will make a identical copies of DNA. Okay, that is by means of replication. The process of making of copies of your DNA is known as a replication by using enzymes. Now formation of RNA, making of RNA from DNA by using enzymes again. Known as a transcription. Making of protein from RNA. By using again enzymes. Process is known as a translation or protein synthesis. These proteins may be either of two types. So whatever proteins they are synthesized, they may serve as a different function. They may act as a structural protein to serve your body. जैसे body के parts के लिए structural protein लगता है. They may be either structural protein or they may be either special protein like enzymes, which may also. This is known as the central dogma of life. Now you can see over here principal basis difference between nucleotide and nucleosides. तो पहले देखो there are different bases like adenine, guanine, cytosine and uracil. So what will be in a nucleoside? पहले ही बोला है साइड मतलब कि साइड कितना होते? दो होते. इसमें कंपोनेंट कितने रहेंगे? दो. साइड मतलब दो. So adenine, okay, so adenine base is there and if sugar is attached to adenine by a glycosidic bond, so it is known as adenosine. तो adenine जो बेस है उसको अगर शुगर अटैच होगा तो उसको बोलेंगे एडेनोसिन मोनोफॉस्फेट. अगर फॉस्फेट उसमें अटैच होगा उसको बोलेंगे एडेनोसिन 5 मोनोफॉस्फेट. एडेनोसिन 5 डैश मोनोफॉस्फेट. तो इज अ न्यूक्लियोटाइड. न्यूक्लियोसाइड क्या रहेगा एडेनिन का? एडेनोसिन. there is a nucleoside of adenine, adenosine 5 dash monophosphate or also known as adenylate. Okay, adenylate or adenylate. That is a nucleotide of adenine. So it is generally abbreviated as AMP, adenine monophosphate. That is adenosine 5 dash monophosphate or adenylate. समझ में आ रहा है आपको? Are you getting? Second one is a guanine. Guanine is the base. तो guanine base अगर होगा तो उसका राइबोन्यूक्लियोटाइड क्या बनेगा? न्यूक्लियोसाइड क्या बनेगा? राइबोन्यूक्लियोसाइड. So guanine plus your sugar, that is known as a guanosine. Guanine plus sugar is known as a guanosine. Okay. Now this guanine plus sugar plus phosphate is known as a guanosine. Monophosphate or guanylate. Okay. So guanosine plus sugar plus phosphate. Guanosine 5 monophosphate or guanylate. Abbreviation is a GMP. Guanine monophosphate. Guanylyl monophosphate. Then cytosine. Third base is cytosine. Cytosine में अगर sugar अटैच होगा, that is known as a ribonucleoside. Okay. So that is known as cytidine. Cytosine plus sugar, cytidine. Now cytosine plus sugar plus phosphate. Known as a cytidine 5 dash monophosphate. 5 dash क्यों बोलेंगे? क्योंकि वहाँ पे जो 5 युक्त कार्बन आइटम है आपका शुगर का, उसके ऊपर फॉस्फेट ग्रुप अटैच होता है. It is also known as a cystidylate. Cystidylate. Okay? So CMP. So uracil plus sugar known as a uridine. Nucleoside is a uridine. Nucleotide is a uridine monophosphate or uridylate. Uridylate or uridylate. So UMP is the abbreviation. This is the ribonucleotides. तो पहले वाले थे आपने देखे. These are the ribonucleotide. Ribonucleotide. They are present in the RNA. यहाँ पे राइबो शुगर है. अभी डीऑक्सीराइबोन्यूक्लियोटाइड ये रहेंगे डीएनए में. ये सब क्या रहेंगे डीएनए में. सो डीएनए में कौन से न्यूक्लियोटाइड रहेंगे, कौन से न्यूक्लियोसाइड रहेंगे. तो एडेनिन अगेन विल फॉर्म अ एडेनिन विथ डीऑक्सीराइबो शुगर, सो नोन एज अ डीऑक्सी एडेनोसिन, दैट इज अ डीऑक्सी न्यूक्लियोसाइड. और उसमें फॉस्फेट ग्रुप ऐड होगा डीऑक्सी एडेनोसिन मोनोफॉस्फेट और डीऑक्सी एडेनिलेट. तो उसको एब्रिविएशन सो दे आर डिनोटेड बाय DAMP. डी मींस डीऑक्सी, डी फॉर डीऑक्सी राइबोस. यहाँ पे डी नहीं है, क्यों, यहाँ पे राइबोस है. सो डीऑक्सी राइबोस DAMP. नाउ guanine, so guanine combined with the deoxyribose sugar will form a deoxyguanosine ribonucleotide. Deoxyguanosine, that is a kind of deoxyribonucleotide. Now nucleotide तीसरा कंपोनेंट ऐड होगा, that is a deoxyguanosine 5 monophosphate or deoxyguanylate. DGMP is the abbreviation. Now cytosine, that will form a deoxyribonucleotide deoxycytidine. Deoxycytidine monophosphate or deoxycystidylate, that is a DMP. Now thymine जब deoxyribose शुगर के साथ में ग्लाइकोसाइडिक बॉन्ड फॉर्म करेगा उसको बोलेंगे deoxythymidine. okay? Deoxythymidine, that is a deoxyribonucleoside. और उसका अगर फॉस्फेट ग्रुप ऐड होगा तो deoxythymidine 5' monophosphate or deoxythymidate. So you can see over here again. So structure of nucleotide. This is the AMP ribonucleotide. ठीक है, यहाँ पे डी नहीं लिखा तो क्या है राइबोन्यूक्लियोटाइड. AMP किसका है देखो, एडेनोसाइन 5' मोनोफॉस्फेट और एडेनिलेट. AMP क्या है, so you can see here adenine base. Adenine base is nothing but the nitrogenous base. It is nothing but the adenine. So adenine with the ribose sugar, okay? And then at a ribose 5 ribose you have phosphate group. So it is known as a adenosine 5 monophosphate or adenylate. Then again TMP means what? Okay, TMP means thymidine monophosphate, okay, or thymidylate. Thymine monophosphate or thymidylate. Okay, this is thymine and ribose sugar, and so these are nothing but the main principal nucleotides and nucleosides. Those are commonly found in the nucleic acids DNA and RNA. Now structure of DNA, इसके ऊपर भी क्वेश्चन आता है, structure of DNA and RNA. First we will see structure of DNA. Now it is nothing but the polymer of your deoxyribonucleotides. This DNA polymer of deoxyribonucleotide. These deoxyribonucleotide they are held together by, मतलब वो वहाँ पे कौन से बॉन्ड फॉर्म होता है, bond between two ribonucleotide, that is nothing but 3' phosphodiester bond. ठीक है, phosphodiester bond मतलब क्या है, एक राइबोन्यूक्लियोटाइड का 3' है और दूसरे राइबोन्यूक्लियोटाइड का 5' है. तो इन दोनों में phosphodiester bond फॉर्म होगा, that is known as a polymer of deoxyribonucleotide held together by 3' 5' phosphodiester bridges in the DNA. So whatever adenine is there is equal to thymine and cytosine is equal to guanine. So total molar equivalence between purines and pyrimidines is there. So DNA के स्ट्रक्चर में क्या आता है, number of purines pyrimidines क्या होते हैं, इक्वल होते हैं, and that was observed by this Chargaff. So that's why it is known as Chargaff's rule. So Irwin Chargaff in 1940, his name is Irwin Chargaff. Irwin मतलब आपका सही तामन करने. Irwin Chargaff 1940 proposed that the DNA equal number of adenine and thymidine residue, AT is equal to A is equal to T, and equal number of guanine and cytosine residue, G is equal to C. This is known as Chargaff's rule. Chargaff's rule will state the molar equivalence between purines and pyrimidines in the structure of DNA. ये रूल आपको याद होना चाहिए क्या है, एकदम सिंपल, 12th में भी आपको हो चुका है. So but they used to ask question on this Chargaff rule. Now DNA structure is nothing but the double stranded helical structure. DNA is nothing but the double stranded helical structure. That is the most common structure found in the nature. DNA का structure कैसा होता है? Double stranded. The double helical structure of DNA was proposed by Watson and Crick. इसके ऊपर भी क्वेश्चन आ चुका है. So James Watson and Francis Crick in 1953 they proposed the double helical structure of DNA. उन्होंने बोला कि DNA के ऊपर आपका जेनेटिक मटेरियल होता है और DNA का स्ट्रक्चर डबल हेलिक्स होता है. And they got a Nobel Prize for that. Now this DNA is made up of two polynucleotide chain. इसमें क्या आएगा कि दो पॉलीन्यूक्लियोटाइड चेन रहेगा. पॉलीन्यूक्लियो मतलब पॉली मतलब क्या है? बोथ साइड. न्यूक्लियोटाइड पॉली डीऑक्सी राइबोन्यूक्लियोटाइड. ये डीऑक्सी बट पॉली डीऑक्सी राइबोन्यूक्लियोटाइड तो उसको बोलेंगे डीएनए. So it is made up of two chains of polydeoxyribonucleotide. Now one chain consists of a genetic information. एक चेन के ऊपर क्या होता है? जेनेटिक इंफॉर्मेशन. सो जेनेटिक इंफॉर्मेशन रिसाइड्स ऑन द टेम्पलेट स्ट्रैंड. उसको बोलेंगे टेम्पलेट स्ट्रैंड. जिसके ऊपर जेनेटिक इंफॉर्मेशन है उसको बोलेंगे टेम्पलेट स्ट्रैंड. Now another is there known as a complementary strand or another known as a complementary strand. Now this template strand and complementary strand they are anti-parallel to each other. Anti-parallel और two strands they are held together by hydrogen bonds. तो ये दोनों स्ट्रैंड एक में के साथ में बॉन्ड किए रहते हैं. इससे हाइड्रोजन बॉन्ड्स. नाउ हाइड्रोजन बॉन्ड्स ऑफ योर बेसिस. pyridine बेस जो होता है pyridine बेस जो होता है उसमें जो हाइड्रोजन बॉन्डिंग होता है. दैट्स व्हाई दिस डीएनए इज अ स्टेबल हेलिकल स्ट्रक्चर. नाउ ईच टर्न ऑफ योर डीएनए इज अबाउट 34 एंगस्ट्रॉम यूनिट. तो ईच टर्न मतलब एक हेलिक का टर्न जो होता है उसका 34 एंगस्ट्रॉम यूनिट का. नाउ इन ईच टर्न देयर आर 10 बेस पेयर्स ऑफ न्यूक्लियोटाइड. एक टर्न में कितना होगा बेस पेयर? 10 बेस पेयर ऑफ न्यूक्लियोटाइड्स. नाउ ईच बेस पेयर इज प्लेस्ड एट अ डिस्टेंस ऑफ 3.4 एंगस्ट्रॉम. मतलब एक टर्न में कितना डिस्टेंस है उसका? एक टर्न का एरिया 34 एंगस्ट्रॉम है. ठीक है. उसमें कितने बेस पेयर रहेंगे? एक इसमें 10 बेस पेयर. तो हर एक बेस पेयर के अंदर हर एक बेस पेयर में डिस्टेंस कितना रहेगा? 3.4 एंगस्ट्रॉम. नाउ टोटल विड्थ ऑफ दिस डबल हेलिक्स इज 20 एंगस्ट्रॉम. क्वेश्चन इसके ऊपर आ सकता है. यू हैव टू रिमेंबर. नाउ टाइप्स ऑफ हेलिकल स्ट्रक्चर ऑफ डीएनए. डीएनए के स्ट्रक्चर के कितने टाइप हैं? देयर आर डिफरेंट टाइप्स. एटलीस्ट 5 टाइप्स आर देयर. Few of them are listed over here which are important. So first one is a B DNA. First one is a B type of DNA or it is known as a bent DNA. Right-handed DNA. Okay? Right-handed DNA. So B DNA is nothing but the right-handed DNA. This is the model proposed by Watson and Crick. क्वेश्चन इसके ऊपर आ चुका है. सो वन ऑफ द फॉलोइंग इज वैटसन एंड क्रिक मॉडल ऑफ डीएनए. ऑप्शन में आएगा आपको बी डीएनए, ए डीएनए, बी डीएनए, ए डीएनए, जेड डीएनए और दूसरा एक ऑप्शन होता है बेंट वगैरह कुछ. बेंट डीएनए, ई. सो रिमेंबर बी टाइप ऑफ डीएनए इज proposed by Watson and Crick. क्वेश्चन आई थिंक जीपीएट में आ चुका है. बी डीएनए इज द राइट हैंडेड डीएनए and it is proposed by Watson and Crick. Then another kind of DNA is there known as A DNA. A DNA is the right-handed DNA. B क्या है? Right-handed है. A B क्या है? Right-handed. Now another kind is there Z DNA. So Z DNA is a left-handed DNA. तो Z type का DNA जो है उसको बोलेंगे left-handed DNA. It is a okay Z DNA is a left-handed DNA and polynucleotide chain in this DNA moves in a somewhat zigzag fashion. ये zigzag fashion में मतलब इसका अरेंजमेंट होता है. That's why Z DNA. Amongst these DNA, B DNA, A DNA, Z DNA, इनमें का सबसे फिजियोलॉजिकली कौन सा कॉमन होता है? सो B DNA is the most prominent under physiological condition. सबसे ज्यादा कौन सा DNA पाया जाता है? B DNA. B DNA is the most prominent under physiological condition. याद रखो. Then other forms of DNA, there are the other forms of DNA like bent DNA, then triple stranded DNA, 4 stranded DNA. पता है ना? जैसे 2 stranded DNA is the most common. उसके बाद में ये भी होते हैं जैसे triple stranded DNA, 4 stranded DNA. तो आपको डिटेल में जाने का जरूरत नहीं. Now this is again very important part. इसके ऊपर भी क्वेश्चन आ चुका है. और denaturation of DNA. What is meant by denaturation of DNA? तो DNA में आपको पता है कि it is a helical structure. 2 strands are there. It is a helical structure. So if any disturbance in the helical structure, a disturbance or destruction of native helical structure of DNA by any means, for example जैसे by using temperature or by using pH के कारण या फिर टेम्परेचर के कारण जो डबल हेलिकल स्ट्रक्चर है डीएनए का उसमें अगर वो हेलिकल स्ट्रक्चर लॉस होता है उसको बोलेंगे डीनेचुरेशन ऑफ डीएनए. So loss of helical structure of DNA is known as a denaturation. Helical structure, okay, Denaturation or helical structure of DNA can be measured by UV स्पेक्ट्रम। आपका DNA इंटैक्ट है या फिर डीनेचर हो चुका है, तो अगर आपका DNA इंटैक्ट है तो उसका लैम्डा मैक्स कम आएगा। अगर आपका DNA डीनेचर हो चुका है उसका लैम्डा मैक्स ज़्यादा आएगा। Because of denatured DNA will release the nucleotides. Nucleotides are nothing but the having conjugation in their DNA. So conjugation they will increase the absolute max. So denatured DNA will show absorbance at a higher level. The phenomenon of increase in the absorbance of purines and pyrimidines following the denaturation is known as a hyperchemistry. Then melting temperature. What is melting temperature or TM? Melting temperature or TM. So it is nothing but the temperature at which 50% of DNA is lost. So melting temperature is nothing but the temperature at half of the helical structure of DNA will be lost. Question इसके ऊपर as it is आ चुका है, क्वेश्चन आ चुका है इसका, ये BDN के ऊपर भी क्वेश्चन आ चुका है, चार्गॉप्स के ऊपर क्वेश्चन आ चुका है, चार्गॉप्स के ऊपर क्वेश्चन आ चुका है। तो यू हैव टू रिमेंबर दिस। और ये तो सिंपल है, इसके ऊपर तो आ भी गए तो आपको आएगा। So double helical structure of DNA was proposed by James Watson and Francis Crick. So you can see here a structure of DNA. So DNA is nothing but the double helical, or it is a polymer of deoxyribonucleotide. It's a polymer of deoxyribonucleotide. ये दो चेन रहेंगे, दो पॉलीमर मतलब is a two polynucleotide chain. Okay. So one chain consists of genetic information known as a template strand, and another one is known as a complementary strand. Template strand is also known as a sense strand, and this complementary is known as a non-sense strand. Now you can see in between these two line the these are nothing but the hydrogen bonds. These are nothing but the hydrogen bonds. Suppose there is one adenine and there is one adenine and thymine. So adenine thymine of two different strand. से मान लीजिए कि यहाँ पे एडेनिन है, यहाँ पे थाइमिन है, इस स्ट्रैंड को, ये दोनों के बीच में क्या रहेगा, हाइड्रोजन बॉन्ड. So this kind of hydrogen bonds they stabilize the DNA structure. Suppose that this is cytosine है और guanine है. So cytosine guanine they will form, this is cytosine, this is guanine, they will form a hydrogen bond. तो अगर यहाँ पे cytosine है, यहाँ पे guanine है, they will form a hydrogen bond. That's why these hydrogen bonds they stabilizes the double helical structure of DNA. Question आ चुका है. The following type of bond stabilizes the double helical structure of DNA. तो उसमें आएगा आपको हाइड्रोजन बॉन्ड, वैन डर वाल्स प्रोसेस, उसमें आएगा फॉस्फोडाइस्टर बॉन्ड, ग्लाइकोसाइड बॉन्ड. मतलब कंफ्यूज करने के लिए आपको पूछेंगे ग्लाइकोसाइड बॉन्ड कहाँ पे बनेगा, बेस पेयर है, बेसिस है एडेनिन pyrimidine and pyrimidine, और शुगर है उसमें ग्लाइकोसाइड बॉन्ड. और बाद में फॉस्फोडाइस्टर बॉन्ड कहाँ पे बनेगा, जो बेस पे जो शुगर मॉलिक्यूल है उसमें जब फॉस्फेट अटैच होगा तो उसमें फॉस्फोडाइस्टर बॉन्ड, उसका और डीएनए का नहीं ज्यादा क्वेश्चन. डीएनए का डबल स्टेबलाइज करने के लिए कौन रिस्पॉन्सिबल है, हाइड्रोजन बॉन्डिंग बिटवीन द बेस पेयर्स ऑफ टू डिफरेंट स्ट्रैंड्स. दोनों स्ट्रैंड्स के बीच में जो, दोनों स्ट्रैंड के ऊपर जो प्यूरिन बेसिस या फिर जो भी नाइट्रोजनस बेस है, वो दोनों बेस के बीच में जो बॉन्डिंग होता है हाइड्रोजन, सो दे विल स्टेबलाइज द डीएनए. नाउ ईच हेलिक्स, दिस पार्ट इज नोन एज अ हेलिक्स. ईच helical structure DNA will take 34 angstrom, okay, 34 angstrom or 3.4 nanometer means 34 angstrom. 34 angstrom means 34 nanometer, 3.4 nanometer. So each helix of DNA will occupy 34 angstrom space. In the each helical, okay, in each groove or in each helix there are 10 base pairs. कितने बेस पेयर आएंगे, 10 बेस पेयर्स, and each base pair is separate. Each base pair is spaced at a distance of 3-4 point angstrom. That is nothing but the 0.34 nanometer. Okay, so each helix will occupy 3.4 nanometer space or 35 angstrom, sorry 34 angstrom unit, and each base pair are placed at a 3.4 angstrom unit or 0.34 nanometer. It is a 3' end of one polynucleotide chain, and it is a 5' end of one polynucleotide. Whatever, like guanine will form a bond with the cytosine, adenine will form a hydrogen bond with the thymine, adenine, like that. This is the structure of your double helical DNA. Now structure of your RNA. So RNA is ribonucleic acid. So it is again a polymer of ribonucleic nucleotides. So what is the RNA? It's a polymer of ribonucleotide held together by 3 dash 3 dash 5 dash phosphodized bond or bridge. Now it has a difference in the sugar. DNA में क्या होता है? डीऑक्सीराइबोस शुगर होता है. RNA में क्या होगा? राइबोस शुगर होगा. And one more, pyrimid में होता है थायमिन, DNA में होता है, and RNA में थायमिन की जगह यूरेसिल होता है. Okay, so it has a difference in the sugar. Instead of thymine, RNA has a uracil. Then usually RNA is a single strand, DNA is a double strand, RNA is single strand. And remember, RNA will not obey Chargaff's rule. Chargaff's rules are not obeyed by RNA, and RNA are susceptible, or they are easily hydrolyzed by base, alkali hydrolysis. They are susceptible to alkali hydrolysis, and RNA will give you color reaction with the orsinol because of the free hydroxyl group of sugar. तो RNA में क्या होता है कि फ्री हाइड्रोक्सिल ग्रुप होता है शुगर का, तो वो ऑर्सिनॉल के साथ में कलर रिएक्शन है. तो टाइप्स ऑफ RNA, तो मेजर 3 टाइप्स आते हैं. मैसेंजर RNA, that is known as mRNA. So totally, if you consider 100% RNA, so out of that 5 to 10% is mRNA. Transfer RNA or tRNA, that is a 10 to 20% of total RNA. Then ribosomal RNA or rRNA, 50 to 80% of total. These are the structure of RNA. We will see the structure of RNA. So this is known as a mRNA. mRNA का structure क्या रहेगा? Linear रहेगा. This is the structure of your rRNA, ribosomal RNA. This one is a tRNA, transfer RNA. Transfer RNA will look like a clover leaf. Okay, इसमें देखो different arms होते हैं. जैसे ये इसका एक, this is a tRNA. tRNA में different arm होते हैं, like anticoulomb arm, then acceptor arm, okay, anticoulomb arm, dourouridine arm, P psi arm, dourouridine arm. So we'll see it in upcoming slide. So DNA का structure तो already हो चुका है. DNA is a double stranded and RNA is a single stranded. DNA में क्या होगा? ये जो थायमिन का बेस है वो होगा, और RNA में वो थायमिन नहीं होगा. तो क्या होगा? यूरासिल होगा. बाकी सब सेम है. बेस में डिफरेंस है, पिरिमिडिन का बेस में डिफरेंस है, एंड सिंगल टाइप्स ऑफ RNA. फर्स्ट वन इज नथिंग बट मैसेंजर RNA. सो एज द नेम इंडिकेट, इसका काम ही है कि मैसेज देना. कौन सा मैसेज देना? जो हेरिडिटरी इंफॉर्मेशन है आपको, उसका मैसेज किसके ऊपर रहेगा? ये mRNA. सो ट्रांसफर ऑफ जेनेटिक इंफॉर्मेशन फ्रॉम जीन्स टू द ribosomes to synthesize the proteins. That is a function of your now eukaryotic mRNA. So there are two kind of cells, prokaryotic and eukaryotic. तो prokaryotes मतलब आपका बैक्टीरिया वगैरह होता है, माइक्रोऑर्गेनिज्म. So in the microorganism, prokaryotic cell is there. In the mammalian there is a eukaryotic cell. So eukaryotic mRNA is capped at a 5' terminal by 7-methyl guanosine. 7-methyl guanosine. तो यहाँ पे एक टर्मिनल रहेगा, जैसे ये ना, ये 3' एंड 5', ये 3' एंड है, 5' एंड के ऊपर ये mRNA के ऊपर क्या रहेगा? ये रहेगा 7-methyl guanosine. Okay, so eukaryotic mRNA is capped at a 5' terminal by 7-methyl guanosine triphosphate. Now this capping prevents the mRNA. क्वेश्चन इसके ऊपर आ चुका है. So what will prevent the hydrolysis of mRNA? So this capping at a 5' terminal, capping of a 7-methyl guanosine phosphate triphosphate, that is the capping that prevents the hydrolysis of this mRNA. Now second type, there is known as so actually this mRNA is not synthesized directly. mRNA का डायरेक्ट mRNA नहीं बनता है आपका बॉडी में. तो mRNA is synthesized as a precursor first. पहले तो क्या होगा? mRNA का प्रिकर्सर बनेगा, और उसका प्रिकर्सर कौन है? ये heterogeneous nuclear RNA, HNRNA, heterogeneous nuclear RNA. So this heterogeneous nuclear RNA will be synthesized in the body first. After modification of this, mRNA will be synthesized. So it is a precursor. It is a precursor for mRNA and other kind of RNA. Now next is a transfer RNA. So transfer RNA is nothing but tRNA. It is also known as soluble RNA, and the structure of this will resemble to clover leaf. So structure पहले देखा है. clover leaf जैसा ये देखिए. For example, you can see over here. So this one is a tRNA. This last one is a tRNA. This one is known as an acceptor arm. Then it is known as the anticodon arm. Anticodon मतलब क्या है? यहाँ पे तीन दिखाए, तीन कोडन मतलब तीन three base pairs are there, which are very complementary to the base pair present in the mRNA. तो इसके नीचे mRNA आएगा तो उसके अपोजिट इसके बेस पेयर है, वो प्रोटीन सिंथेसिस के लिए mRNA और this tRNA they comes together. Protein synthesis takes place in the ribosome. So you see it. Your tRNA has a different transfer RNA. Okay, transfer RNA. What is the function? Transfer of amino acid. Transfer of amino acid to mRNA for the protein synthesis. Now tRNA consists of four arms. First arm is nothing but the acceptor arm, which is having a cap at a 5-3' It is a CCA. तो इसके ऊपर वाला जो पार्ट है एक्सेप्टर आर्म उसमें CCA सीक्वेंस रहेगा. C मतलब साइटोसिन साइटोसिन एडेनिन, ये सीक्वेंस रहेगा. दैट इज अ एक्सेप्टर फॉर अमीनो एसिड. तो वो अमीनो एसिड को पकड़ के लेके आएगा. तो देयर आर यू नो दैट देयर आर 20 स्टैंडर्ड अमीनो एसिड्स. तो नेचर में कितना है 20 स्टैंडर्ड्स अमीनो एसिड्स. तो एमआरएनए भी कितने रहेंगे उसके लिए 20 रहेंगे. CCA एक हर एक अमीनो एसिड के लिए एक आरएनए रहेगा. So there are 20 amino acids and therefore there are 20 respective tRNAs. ठीक है. Now amino acids they are attached to a acceptor. आप बोलेंगे यहाँ पे amino acid क्यों attach करना है? For protein synthesis. What is protein? They are nothing but the polymer of amino acid. So polymer तो add करना पड़ेगा ना उसमें. So that's why acceptor arm they will carry amino acid. Okay. Now second arm is known as anticodon arm. Anticodon मतलब codon जहाँ पे भी 3 base pair का sequence रहेगा, which are complementary to the base pair present in the mRNA. So containing 3 specific anticodon responsible for the recognition of triplet codon on the mRNA. Now third arm is nothing but the D arm. D arm because it is given a name D because of the presence of dihydrouridine. ये D arm में dihydrouridine होता है, उसके लिए उसको D arm बोलते हैं. Now fourth one is the T-Psi-C arm. So it consists of a sequence of pseudouridine. Pseudouridine रहेगा इसलिए T-Psi-C arm. Now next one is a variable arm. Fifth arm is a variable arm. So because of the variability, कुछ RNA में ये 10 to 20 base pair का रहेगा, कुछ RNA में 20 to 60 base pair का रहेगा. So depending on that, tRNA is classified as a class 1 and class 2. We can see what this is known as acceptor arm. This one is known as a D arm or dihydroxyuridine arm. This one is known as a TYC arm, T-Psi-Y arm, so that is known as pseudouridine arm, and this one is known as a variable arm. Short one is variable. This is the structure of tRNA, which resembles to, or which will look like a clover leaf. Clover का plant है, उसका पत्ता जो होता है उस तरीके से वो दिखता है. Now next one is a ribosomal RNA, called as a rRNA. So it is nothing but it will provide a structural framework for ribosomes. GPAT 2019 में क्वेश्चन आया था इसके ऊपर. स्मॉल न्यूक्लियर आरएनए SNRNA, स्मॉल न्यूक्लियर आरएनए SNRNA. So what is the function of it? It involves the processing of mRNA. Small nuclear RNA SNRNA involved in the processing of mRNA. mRNA का प्रोसेसिंग करेगा. ये कुछ गलती है सिंथेसाइज किया mRNA, इसमें कुछ मिस्टेक है, कुछ एरर्स आ गया तो वो करेक्ट करेगा. Small nuclear RNA SNRNA. GPAT 2019 में क्वेश्चन आ चुका है. Then next one is the small nucleolar. So ये क्या था? न्यूक्लियर था. Small nuclear RNA SNRNA और small nucleolar न्यूक्लियोलस में रहेगा. ये न्यूक्लियस में रहेगा, ये रहेगा न्यूक्लियोलस में. SNRNA. So again it has the same plays a key role in the processing of RNA molecules, processing of mRNA molecules. And again, okay, small cytoplasmic RNA SORNA. So again it has a function of selection of proteins for export. जैसे एक बार प्रोटीन सिंथेसिस हो गया तो प्रोटीन का एक्सपोर्ट भी करना है ना, तो वहाँ पे थोड़ी रखना है, तो उसको बाकी काम के लिए निकालना है. तो SORNA और small cytoplasmic RNA involves the it is involved in the selection of protein for export. Now next one is the transfer messenger RNA. tmRNA क्या है काम उसका? Mostly present in the bacteria and function as an addition of short peptide tags. छोटा सा पेप्टाइड आ रखेगा टू द प्रोटीन टू फैसिलिटेट द डिग्रेडेशन ऑफ इनकरेक्टली सिंथेसाइज्ड प्रोटीन। जब प्रोटीन सिंथेसिस होता है, ट्रांसलेशन जब होता है, तो कभी कभी क्या है कि इनकरेक्ट प्रोटीन सिंथेसिस होता है। तो अगर वो हो गया, सो व्हाट इज नेक्स्ट स्टेप? So you have to degrade that protein. So for that, this tmRNA, transfer messenger RNA, so mostly present in the bacteria, and it adds in some peptide tag, मतलब छोटा सा पेप्टाइड ऐड करेगा इस प्रोटीन के ऊपर। कौन सा प्रोटीन? जो एक्यूरेटली सिंथेसाइज नहीं हुआ है उसके ऊपर वो ऐड करेगा छोटा सा टैग, तो वो बाद में उसका डिस्ट्रक्शन हो जाएगा। टाइप्स ऑफ आरएनए, मैसेंजर आरएनए अगेन एब्रिविएटेड एज अ mRNA, messenger RNA, mRNA. What is the function? Transfer of genetic material from, so transfer of genetic information from genes to ribosome to synthesize the protein. Heteronuclear, heterogeneous nuclear RNA, HNRNA, ये आपने देखा है already. We have seen this as it is given in the Satyanarayana. Now actually this nucleic acid is again very vast chapter, and there are a lot of things there are a lot of things to explain from this chapter, बट उतना ज़रूरत नहीं है आपको। सो टेकिंग ओनली इन शॉर्ट में ले रहा हूँ। सो आपको एक डिसीज आता है, देखो गाउट, आर्थराइटिस एंड गाउट इज अ डिसीज ऑफ योर जॉइंट्स, गाउट्स। सो इट इज अ पर्टिकुलरली मेटाबॉलिक डिसऑर्डर ऑफ योर प्यूरिन्स। प्यूरिन का मेटाबॉलिज्म अगर ज़्यादा होता है, तो व्हेन देयर इज अ इंक्रीज्ड यूरिक एसिड इन द, दैट यूरिक एसिड गेट डिपॉजिटेड इन द joints, particularly synovial joints. Okay, so uric acid, that increased uric acid of your body will react with the different cations like sodium, calcium, potassium, and they will form a urate, sodium urate, calcium urate, potassium urate, and that crystal will be deposited in the your joints. Joints के ऊपर वहाँ पे deposit होता है, and that crystal causes the swelling, inflammation, and known as a gouty arthritis. That is due to the increased metabolism of your purines. Okay, तो प्यूरिन में क्या आता है? एडेनिन, एडेनिन मोनोफॉस्फेट, इट विल बी टेकन बाय एडेनिन मोनोफॉस्फेट डीअमाइनेज। डीअमाइनेज मतलब अमोनिया रिलीज हो गया, सो रिलीज ऑफ अमोनिया एंड प्रोडक्शन ऑफ इनोसिन मोनोफॉस्फेट, so it will be converted to inosine by removal of phosphate. Then inosine will be converted to hypoxanthine. Hypoxanthine will be converted to xanthine by xanthine oxidase. This is very important step. Xanthine will be converted to uric acid by xanthine oxidase. And this uric acid, if increased in the body, it reacts with the cations from sodium urate, calcium urate, or potassium urate, get deposited in the bone, get deposited into synovial joints and causes arthritis and gouty arthritis. Now that's why xanthine oxidase inhibitor they are used. Xanthine oxidase inhibitor कौन है? Tell me the example of xanthine oxidase inhibitor. आपने B.Pharm में क्या किया? आपने फार्माकोलॉजी में पढ़ा होगा। xanthine oxidase inhibitor. Anybody? Xanthine oxidase inhibitor drug. So remember allopurinol. Allopurinol, ALLOPURINOL. Allopurinol is a xanthine oxidase inhibitor. So इसको इनहिबिट किया तो xanthine नहीं बनेगा, xanthine नहीं बना तो यूरिक एसिड बनेगा, यूरिक एसिड का लेवल कम हो जाएगा और गाउट से आपको छुटकारा मिल जाएगा। So xanthine oxidase inhibitor they are used in the treatment of gout. So gout आपको रिलेट कर सके इसलिए ये लिए, अदरवाइज इंडिविजुअल प्यूरिन है, इंडिविजुअल प्यूरिन्स, इंडिविजुअल प्यूरिमिडीन्स, सबका मेटाबॉलिज्म होता है. It is given in the books, but I don't think that is important. So it is important, so that's why I have taken. Now you can see over here. So what is your DNA? DNA is nothing but the polymer of your deoxyribonucleotides. Polymer. So for example, this is a one nucleotide. So this is thymine base. So that this is sugar part phosphate. So it is a nucleotide. So phosphate is attached to a 5th carbon atom of your sugar. So 5' phosphate end. To this nucleotide, another nucleotide is attached by phosphodiester. ये देखो इसमें यहाँ पे इसका कौन सा है? साइटोसिन. कोई साइटोसिन, साइटोसिन में इसका phosphate group यहाँ पे attach हो गया. By using this phosphate will make a bond with the third carbon atom of this pentose sugar. So that's why it is known as 3' 5' like this. ओके, तो ये चेन का क्या रहेगा? 5' एंड जो है वो ओपन रहेगा, और ऐसे ही दूसरे चेन का क्या रहेगा? 3' एंड ओपन रहेगा. सो 5' 3' कॉम्प्लीमेंट्री स्ट्रैंड विल हैव 3' 5' like so remember these two nucleotides, they are combined or they are bound together by phosphodiester bond. Yeah, देखो phosphodiester. ROR is known as ester. Then again ROR diester. Phosphate is there. So phosphodiester bond. These three nucleotides they held together by टू फॉस्फोडाइस्टर तीन न्यूक्लियोटाइड से तो उसमें दो फॉस्फोडाइस्टर बॉन्ड रहेंगे जैसे 10 न्यूक्लियोटाइड रहेंगे तो उसमें 9 फॉस्फोडाइस्टर बॉन्ड रहेंगे तो आपको ये भी क्वेश्चन आता है कि ए पेप्टाइड कंटेनिंग 105 nucleotides. A DNA containing 105 nucleotides. So how many phosphodiesterase bonds are present in that? So obviously one less. So 105 बेस पे रहे तो उसमें 104 बॉन्ड रहेंगे वन लेस मतलब यहाँ पे देखो ना दो न्यूक्लियोटाइड है अगर तीन है थ्री न्यूक्लियोटाइड्स आर देयर तो यू हैव टू बॉन्ड्स you have two phosphodiester bonds. Now DNA replication is there, so we'll take it tomorrow.